Welcome those on the phone. Sorry you're muted. It's not that we don't want to hear your lovely voices. We just felt that it would be easier for a webinar. There's a chat line open, so feel free to post your questions should you have any. We'll also save some time at the end for questions, and we will we'll circulate the slides afterwards so everyone has it, and we're recording this if you miss anything. So today's our second installment of the Type 2 Diabetes Knowledge Portal webinar, and interesting.
Yeah, is it animated? Maybe.
Nope,
Do that again.
we're not pulling anything.
That's weird. Okay, well.
Well, anyway, sorry for that, guys. Things are a little slow on the slides today. Interesting. I am not the only one talking, and there's Ben. We also have Maria speaking. So today, the last time we all talked, we gave you an overview of the portal, the software stack, how it's built, the data underlying it, how we built it, and why we built it. Today, we're going to focus on a deeper dive. Today, we're going to focus on gene-specific resources in the knowledge portal. So Maria is going to take you through that, and I'll talk a little bit about that in a second. And then Ben's going to give you a sneak preview of what's to come. And then I thought I'd start with a very simple reminder of our motivation as to why we did all this. And it's simply rooted in human genetics, in that human genetics is a powerful tool or utility in understanding disease biology and informing human health. And it is also, what human genetics is really natural perturbational experiments that we can observe and test collections of variants, or these perturbations, across collections of phenotypes and produce effect sizes, which help give us insight to the direction, the magnitude, and the effect of particular variants on phenotypes of interest. And for the last 10 to 15 years, I would wager, we've been in this interesting tension, I would say, between great opportunity and challenge in human genetics, which is the great opportunity coming from the fact that you have a motivated research community, specifically type 2 diabetes here, who is interested in generating large-scale human genetics data, sharing it, having the best practices, having the best types of methods and ways of handling the data, representing the data amongst themselves. And that leveraging and that opportunity has yielded a lot of knowledge about human genetics in the space of type 2 diabetes, with over 243 loci identified for type 2 diabetes. That yields 403 independent signals. But the challenge there is where it gets complicated. Many reasons. One is few of these signals have been translated into specific genes or variants. In many cases, we don't know the molecular and cellular function at these variants. Moreover, even the targets or the directionality of the relationship hasn't been challenged. So why do we have these challenges? Well, it's because research is needed in many areas. And I love this slide by Mark McCarthy, which kind of gives you the complexity of which it's important to bring together different lines of evidence, different computational tools, data sets, inference, leveraging the human genetics data. And so our goal with the knowledge portal is very, very simple, is to help to sort of move the opportunity into more challenge space, in the sense that we felt that making genetic data and related genomic data broadly accessible, but moreover useful, could have a significant impact on our ability to understand and treat human disease. So with that, we've built a research and software platform to organize human genetics data and supporting relevant omics data and present these analyses to non-experts. And that's what the portal is. And at the last webinar, we reviewed really basically the features of what you can do with the knowledge portal, which allow you to query data sets by a trait or a data type interactively, allow you to view genome-wide association statistics for a given phenotype, view associations for a specific gene or locus, layered with customized visualizations and annotations, and allow you to customize criteria for variant selection, allow you to view variants across data sets, and finally, dynamically execute custom analyses. So those are the big features. And what are the big data? We mentioned this last time in detail, but really you have 180 traits to query at your fingertips, over 75 data sets, and they represent the diabetes genetics community, public GWAS data sets of value not just for diabetes, but for cardiometabolic traits, psychiatric traits even, AMP-funded data sets. So the AMP T2D partnership funds the knowledge portal and also funds a generation of more data for type 2 diabetes and its complications. That you have at your fingertips. You also have disease agnostic resources, and moreover, biobanks are coming into the portal in the coming years, specifically UK Biobank. What's it powered by? Well, it's powered by a modular and flexible software platform, which we talked about last time. But what's important about this, there are 4 pieces that are really important here. One is the data ingest. We can take individual level data, raw data, and analyze it and represent it in the portal. We can also bring in association statistics from anywhere. Summary level statistics are all we need to represent and aggregate human genetics data. I'm noticing there's a delay on the slides. Is that true? So I apologize because my computer's got a significant delay. I apologize. So I'll go a little slower. We also allow the support of the integration of omics data. So if you have association statistics, we can run computational methods to allow you to bring supporting lines of evidence to bear on these GWAS data sets. And additionally, one other component is federation, which is we are not alone at the Broad, place to house and warehouse and represent the data. We need to partner with people who are experts in different domains where the data reside. And this is federation both at the University of Southern San Diego and at EBI. Finally, you're to represent in the portal. And so these components make a software platform. So what will you learn today? So today, you're going to learn how to access the best of what we know today about predicted T2D effector genes and what we know about the supporting evidence for those. You're going to learn how to query integrated human genetics data with layered annotations for your favorite gene. You're also going to learn how you might prioritize variants for functional study within your favorite gene or region. And then finally, you'll learn how to access computational methods to assist in gene prioritization. That's the landscape today. At our future webinar, we plan to do these quarterly, I believe. There's some really nice other tools that we want to really spend some time on. One is LocusZoom. I think that's a tool that's used outside of the portal. Actually, it's a standalone tool from the University of Michigan. We're going to spend some time on that in a future webinar. We're also going to invite our colleagues, Kyle Galton, who runs the Diabetes Epigenome Atlas, because this is a connected federated node of the knowledge portal. And this will allow us to sort of yield toward that and see how it's connected to the portal, how it helps us enhance our understanding of genes and variants to prioritize. We'll also have new complication data for us to represent to you. We'll also have more methods run on GWAS data sets, and we'll talk about those. And then ultimately, actually, we're going to move to a place where you can interact with the types of diabetes knowledge portal stack in a command line setting via APIs, which is something people have been really excited about. So we'll talk about that at future webinars. But with that, I'll turn it over to Maria, and she will walk you through the gene-specific resources.
Okay. So before I start, I just wanted to remind us all that the T2D portal is part of the Knowledge Portal Network, which has several other portals for complex diseases. And some of what I'll talk about today is currently only in the T2D portal, but we're hoping to expand these interfaces to all the portals and all the phenotypes eventually. So that's coming. And we have a homepage for the Knowledge Portal Network, kp4cd.org, where you can connect to all the portals. And if any of you want to follow along, you probably already know this, I hope, but to log on to the portals, you need a Google account. And so you can log on that way. Okay, and so this is what was left out of the previous slide for some reason, but this is what I'm going to talk about, gene-specific resources. And I'll start off talking about a couple of types of information that are just available for T2D, and then talk about some more general features of the gene page. Okay, so our overall goal, as Noelle just mentioned, is to accelerate the translation of genetic information into knowledge about the specifics of human biology, about proteins, pathways, and networks, and then ultimately to translate that knowledge into new therapeutic or diagnostic opportunities to help patients. And so, of course, it's not only us, but the whole T2D research community has been working on this type of thing, and a lot of different approaches have been developed. And in particular, in Mark McCarthy's group, this figure came from him, he and Anuba Mahajan have been working on a method for prioritizing potential T2D effector genes, for taking all of this enormous, massive genetics data, along with other data that have been accumulated, and figuring out a way to give weight to different types of evidence and create a score, and then be able to categorize genes by the likelihood that they are important to T2D. And so there are three types of evidence that they considered to do this scoring system. First of all is genetic evidence, genetic associations. Second is functional evidence, such as evidence about regulation of genes being important in certain tissues that they're such as pancreatic islets that are important to diabetes. The third type of evidence is perturbational evidence. So what happens if you knock out a gene? What type of mutant phenotype do you get? Or if you knock out the homolog of a human gene in a model organism. So I'm going to go over these three types of evidence at kind of a high level. All of the details of this scoring system are documented in a downloadable PDF you can get from the portal. but I just want to give you an idea of what types of evidence they considered in this system. So for genetic evidence, they looked to see if, so the posterior probability of coding variants in particular genes from credible sets from the DIAMANTE European GWAS, which was a huge GWAS of almost 1 million people. And they also looked at the posterior probability of coding variants in credible sets from the X-Tex T2D, which was an exome array study. And they considered the gene level association scores from the AMP T2D genes exome sequence analysis. And I'll come back to these scores because we present them in an additional way on the portal. They also looked to see whether genes were involved in whether monogenic mutations could confer diabetes or related phenotypes. And they looked at the literature to see if there were any focused genetic studies that had been done for individual genes. For regulatory evidence, they looked to see whether cis-EQTLs had been found that influence expression of the gene, particularly in pancreatic islets, but also in other T2D relevant tissues like fat, muscle, and liver. They also looked to see whether capture C or high C experiments had shown that a T2D associated variant contacts a gene in islets, or whether a T2D associated variant affects chromatin accessibility in or near the gene in islets again. They also considered one study, a published study, where they looked to see whether genes were glucose regulated in islet tissue. And so in this method, they assigned a score if a gene was glucose regulated in pancreatic islets. Finally, the perturbation evidence. They looked at a study from a large-scale study from Anna Gloin's lab, where they did RNA interference to silence a set of human genes, a candidate set. And if that silencing conferred T2D relevant phenotypes, that was considered to contribute to these scores. They also looked at a bunch of model organism databases to look at the mutant phenotypes conferred by mutation of the homologs of a human gene. And if it conferred T2D relevant phenotypes, that contributed to the score. And finally, they looked in the literature to see if any focused mutant phenotype studies had been done for these individual genes. Putting all this together, they developed a scoring system, and were able to categorize each gene by the likelihood that it's an effector gene. And there are about, I think, 130 genes total on this list. And so our job at the portal was to take this list and the evidence and make them accessible to researchers everywhere, and also try to make it understandable so that people would, be able to see what the supporting evidence was. So first of all, this is how you navigate to this list. And I should say we're working on redesigning the homepage and making it even easier to navigate, so that this is going to change, but it'll be obvious how to find this list. So right now, there's a link in the upper menu bar, predicted T2D effectors, and then also in the Explore by Phenotype section, there's a radio button to click to view predicted T2D effector genes. So either of those ways will get you to a page with a big table on it. This is only obviously a very small part of the table, but the table that you initially see has just 5 columns. It has a column for gene names, and those are linked to their gene pages in the portal. And on the leftmost column shows the combined prediction. So this is the prediction from all of the evidence, the sort of bottom line prediction of likelihood that this is a T2D effector. And then there are 3 columns, each showing a score for that the gene got based on the evidence for genetic, regulatory, or perturbation evidence. So that's what you see at first, but there's a lot more to explore here. So just above this on the page, there's an interactive graphic that explains to you how these evidence types were combined to make the overall score. And when you click on, so at the top of this graphic, there are the list of the categories, causal, strong, etc. When you click on any of those, the graphic below changes to show you what went into that classification. So for example, on the left, we're looking at causal, and you can see that only genes in which there was a causal coding variant were classified as causal genes. On the right, we're looking at moderate genes, and in this case, the combined evidence is much more complicated. There are 4 ways you could classify a gene as moderate. And basically, they combine evidence, weaker evidence in the second and third tiers, but it must be from at least 2 different categories between coding, regulatory, and perturbation. So you can explore all of the classifications like this. And as I said, there is complete documentation. So here's the top of the page with some explanatory text and that interactive graphic, and then there's a link to download really extensive documentation. Okay, so going back to the table itself, each of the evidence types columns expands to show the underlying evidence. So this is, for example, the combined genetic evidence. If you click on the little arrows in the right-hand corner of the header, you can see, expand it to see the types of evidence that went into that classification. So for example, for genetic evidence, as I mentioned, there's a column for the GWAS DIAMANTE evidence. There's one for the X-tex T2D exome array evidence, one for the burden test evidence from the exome sequences, any monogenic associations, and any other studies that have been done that bear on the genetic evidence. For each of these columns, there's a help button that just gives a short summary of what's in that column, just to help you get oriented. And also within the table, wherever the information in a cell is underlined and there's a little magnifying glass, that means that you can click it to see more. And these are data that are drawn dynamically from our database. So for example, on the left here, we've clicked on an entry in the GWAS column, and now we see all of the variants that went into that credible set that supplied the evidence for the score in that column. So there's a lot to explore here. I'll just mention briefly that these classifications are also shown in the gene focus table, and I'll come to this later. I'm going to spend some time explaining this table, but it's accessible off the gene page, and this is the only other place at the moment in the portal where these effector gene classifications are shown. Okay, so that is the effector list, and that we have a lot of plans for it. We obviously, it's not going to be just a static list because the underlying evidence is going to grow and change, and so we want to develop a method to keep up with it. Anuba and Mark did much of this work really manually to curate all this information, and that's probably not sustainable in the long run. Certainly, I don't think they're going to be able to keep it up. So we have to figure out ways to sustain this, and figuring out more algorithms for gathering the evidence and evaluating it. So that's going to be a major focus for us, and we do want to update this list at least every quarter. So we hope that this, that already this is a really useful tool for T2D researchers to help direct them and prioritize them to between genes that they may be interested in, and we hope to make it even more useful as we go forward. Okay, so you have any chat questions or anything, or no? Okay, good. We'll have a question and answer period at the end also, so happy to answer questions then. All right, now I'm going to talk about gene level associations, T2D association scores that we've made available in the portal. So I'm sure you're all aware that there's a recent paper with our own Jason Flanagan as first author from worldwide researchers with the largest disease-specific collection of exome sequences to date. And in the portal, we call this the AMP T2D Genes Exome Sequence Analysis Dataset. And that analysis generated both single variant associations and gene level associations. So the single variant associations are integrated into the portal just like all our other associations are. But then the study also produced gene level associations, which are really valuable given that a lot of rare variants were uncovered by the exome sequence analysis. So we hadn't previously presented gene level associations in the portal. So we've made a new interface to look at these, and it's accessible from the home page under this Explore by Phenotype menu. Again, if you click View Gene Level T2D Associations, you can get to that page. And so the page is a table, and it has gene names, which are linked to their gene pages, and other informations about the genes and the number of variants that went into the analysis, and allele counts and so forth. You can change the variant filter pull down here to see scores that were generated using different sets of variants or different aggregation tests. So I'll show you that. So when you pull down this menu, you can see all the options there are. You can choose to restrict the set of variants going into the test to those that were predicted to be deleterious by several different numbers of methods. You can choose the ones that were really, really bad, or just the ones that you know, were not predicted so deleterious, but they have a particular math. There are lots of choices here. There are also two different aggregation test results that you can choose. So when you choose these, the table it redraws, and you can see the results from those. And so this is only available right now for the type 2 diabetes phenotype, but Peter is here. Yes, Peter and Jason are working on associations from this study for many, many quantitative traits. And those will be available, both the single variant associations and also as gene level scores. So there will be gene level scores like this for many phenotypes besides type 2 diabetes in the future. All right, so now I want to talk a little bit about some gene-specific resources that are on the gene page. Okay, so this is just a reminder of how to get to the gene page in the first place. On the home page, there's an Explore by Gene, Variant, or Region box, and if you just start typing a gene name in there, you'll be presented with a list of choices, and you can pick one and get to that gene page. Also, I hope this is review, but just to remind you, the gene page integrates information across a region, not just the coding sequence of the gene, but 100 kb of flanking sequences on either side. And the traffic light at the top is meant to give a quick summary of associations across this whole region. And this traffic light remains constant no matter what else you do with the rest of the gene page, as I'll show you. So green, of course, means there's a strong association for some phenotype. You know, at least one of the 180 that we have. And yellow means moderate association, and red means there's no association. So just below this is a section that we call the phenotype bubbles. They have the same color coding, so the green ones have a strong association, and so forth. And when you click one of these bubbles, it changes everything else lower down on the page to pertain to that phenotype, but it does not change the traffic light. Below this section, there are 4 tabs on the gene page, and they also show what phenotype you're looking at, just to make it clear. Last webinar, I talked some about the top variants and the high impact variants tab, so I'm not going to cover those today. I'm going to focus in a little bit on the credible sets tab and the genes and region tab then. Okay, so credible sets can be really helpful in providing an immediate suggestion of what might be the causal variant for a locus. So credible sets are sets of variants that together have a defined probability of including the causal variant. It's usually 95%, I think. And that they are calculated by researchers doing fine mapping using GWAS datasets. So here's just an example. These are associations across a region of the MTNR1B gene, and you can see that all the top variants, there are many, many variants that are very significantly associated with T2D. But then after the credible set calculation, and this point is kind of creeping off the plot here, but the credible set includes only one variant in this case. So immediately this kind of narrows down the field and points to a variant that could be the one to focus on. So that's why credible sets are important. And so for type 2 diabetes, we include the credible sets from the DIAMANTE GWAS study. So this was a very large GWAS study of almost 1 million participants. And this table shows just part of the credible sets tab, but I just wanted to explain this table because this is really the heart of the page of the tab. Going across the top, these are the variants that are in the credible set. And then below each if any of these variants were located in a coding, splice site, UTR, or promoter regions, the box would be shaded in. As it happens, none of these are. But then their posterior probabilities are shown, and also their p-values. And a really useful thing about this display is that it integrates epigenomic data from the Diabetes Genome Atlas. And these are chromatin states that show the regulatory potential of these regions. And so if a variant overlaps with one of these chromatin states that are listed here, that the box is colored in. So all these orange boxes means that the variant is located within an active enhancer chromatin state. And then the tissues in which those states are found are listed here. So you can immediately see if a variant of interest might have regulatory potential in a tissue of interest. So we have those credible sets for type 2 diabetes. We don't have researcher calculated credible sets for other phenotypes at the moment, although we're certainly planning on adding those. But in the meantime, we have an automatic calculated credible set and this is a LocusZoom module from our friends at the University of Michigan. And so this just does the calculation to create a credible set. And there is a caveat that the calculation assumes that there is one variant in the interval that is associated with a selected trait. And so obviously that's not always the case. So you have to be aware of that. But still, in the absence of other credible sets, this can be a very useful tool to help focus in on variants of interest. All right, so I think the next, okay, now I want to talk about this new table. So everything I've talked about so far really is part of this workflow of going from genetic information, integrating lots of other types of information, and moving to knowledge about biology and disease. And this new, so we've added a new approach to the portal that takes all these different types of information and integrates them and presents them to researchers to help them prioritize genes in a region that may be good directions for further research. This table and tab, the name is kind of evolving, and we haven't totally settled on one. We call it sometimes the Gene Prioritization Toolkit. The tab on the gene page is called Genes in Region. Recently, Ben came up with an acronym that I like a lot because it's easy to say and remember, and that is FOCUS, Find Orthogonal Computational Support. So I'll be calling this the Gene Focus table, I think. So to do this table, to present this information, we had to do a lot of background work that our researchers have been working, our engineers have been working really hard on for a while now. To be able to access all the data in the portal and run it through computational methods, different methods, and run them quickly and in a versatile way, we had to store the data in a different way. And so the data are stored now in a graph database, a Neo4j database, that stores connections between different data types, many different data types such as genetic associations, epigenomic annotations, EQTLs, tissue-specific expression, phenotypes, genes, of course. All those connections are in this database, and that makes it possible to implement many different computational methods that integrate some or various combinations of these types of data to come up with predictions about likelihood of genes being involved in a tissue or in a phenotype. Many different predictions about different aspects of these connections. Some of the methods that we're running right now are Colloc, eCaviar, Metascan, and Depict. And I'm not going to try to explain these methods because it would take a lot of time, and I'm not the best person to do that in any case, but they are documented in our downloadable documentation, of course, with links to the original papers and the software, I should say. These were all developed by different groups around the world. And we are actively looking into other methods and planning to add them as well. So we've stored the data in this new way. We've run these methods and come up with predictions from each of them. And now we're presenting them on this Genes and Region tab on the gene page in the focus table. And so I'm going to spend a little time taking you through how this table works. So when you look at it first, here's the default view. So the genes in the region are going across the top. And those are in that region of the gene you started with plus or minus 100 kb. These are the genes that are in that region. And then the methods are the rows here. So there are the methods that I mentioned. And in addition, we include the gene level association scores from the T2D genes exome sequence analysis study, 2 different burden test methods. And we also include mouse knockout phenotypes. So if any phenotypes conferred by knockout of the mouse homolog of that gene. And as I mentioned before, if a gene is included in the effector gene list, we also put its classification here in this table. The help buttons give you a short summary of what you're looking at for all of these. Results, so the results from each of these methods for each of these genes are shown in the boxes, and they're color-coded by significance. So darker color means more significant. But note that depending on what the output is, significance may be a larger or a smaller number. So for example, for a metascan, the output is a p-value, so smaller is more significant. For colloq, the output is a colocalization posterior probability, so higher number is more significant. So that's why there's that disconnect. But in any case, the darker color means more significant. Okay, this table is also really interactive. You can remove columns if you want. For example, in this example, this rightmost column has practically nothing in it. We're probably not interested in it, so we could exit out to get rid of it. And you can also drag columns if you want to put two genes next to each other to better compare just those 2 genes. You can also, of course, sort the columns by just clicking in the column header. All of the results that are in the cells are linked to details. So when you click on them, a pop-up window will show you the output of that method for that gene. So for example, here's a colloq output, which shows tissues and gives posterior probabilities. And yeah, as I just said, the columns can be reordered or sorted. So in this case, I've removed that mostly empty column. And then the whole table can be transposed. So this transpose button at the top pivots the table so that now the methods are columns and the genes are rows. And this is an easy way to compare across methods. I mean, obviously, I'm sorry, not across methods. The output across methods cannot be compared because the methods use different inputs and they come up with completely different types of results. But across one method, you can compare genes. So this, transposing the table like this, gives you a straightforward way to compare across genes, and you can sort by the score in one method to see which gene did best. There's also a different view of the table, a records view, which is you can get from this button up here. And this simply shows the number of records that were generated from each of these methods. And it's color-coded again. In this case, darker color means fewer records. And our thinking here was that, for example, if a method comes up with a list of 15 tissues in which this gene might be important, maybe that's not so interesting as if it came up with just one tissue that, it could be pancreatic islets. I don't know, we'd be interested in feedback on the usefulness of this view. Not totally sure that it's going to be too helpful, but maybe it will be. But we'd love to hear not only about this, of course, but any aspect of anything I've talked about. We would always want to hear your feedback. Okay, future directions for this table. As I said, we just want to add more methods and more annotations, make it more comprehensive. I think we're going to want to make this a standalone interface too, not so that you don't have to go to a particular gene page to find it, but you can just go to the interface, set your own region of interest, and then see the results. And then the concept of this type of table containing the results of these types of methods, we are expanding to tables for tissues and variants. And Ben is now going to demonstrate, we actually have a working version of the tissue focus table, and Ben is going to demonstrate that for us.
Hello, everybody. I'm going to now give a quick demonstration of one of the newer pieces of functionality that we've integrated into the portal, and this is the tissue enrichment screen, also known as the tissue focus table. And as Maria showed very nicely, we have this notion of a gene focus table, which should allow you to use different computational techniques to compare across different genes. And the notion of this table is that it should allow you to use different computational techniques to compare across different tissues. So if you click on this link at the top of the screen, you will by default end up looking at a screen that is built around the T2D trait. I'll switch to another trait later. So the basic idea of this table, as with the gene focus table that Maria was showing earlier, is that we're starting with GWAS information. We're starting with a GWAS signal that identifies a locus or a series of loci, and from those we identify variants, and then we can use those variants to perform various calculations. So this one that I'm showing in the left-hand column is an LD score regression. So the idea here is you take that set of variants relevant to T2D and then undertake an LD score regression stratified by tissue, and you end up with p-values that rate the different tissues. And so if I then sort by these p-values, you can see that the sort of tissues that we might expect or we might hope to show up at the top, like pancreas and liver. Some others as well. There's some caveats to using this table, but I'll save those for later. Note that Maria also made some very nice help text for all of these different methods, and that provides additional context. Now, a second method that we're using is Gregor, and the outline of the way that Gregor works is, again, you start with a series of variants, and then those variants overlap different annotations. And then we can look in those annotations across different tissues and see how tissues are enriched compared to one another. And so in this case, what we've done are to use the annotations. Here, I'll sort by Gregor p-values. We're using the Chrome HMM chromatid state predictions, and we are coming up with these enrichment scores. And as you can see, again, the tissues that are at the top of the list are ones that make sense for T2D. Not all of them, but some of them. And then for each of these scores, you can see which different annotations are providing the particular enrichment score. And what we do here is just take the best value, and that's the one that I present in the box. So we could have used any of the Chrome HMM states to come up with a score, but what we're doing here is using any predictions of enhancers. And if you think that there's a different set of chromatin states that would provide information, then you can change them. So here is the list of all of the different Chrome HMM states that we have. And if I change some of these, let's say I remove some from the list, then that annotation is no longer contributing to the enrichment of the tissue. So keep an eye on the scores in the right-hand column as I remove some of these annotations. And you'll see that some of the scores change. And in this case, that means that for those scores that changed, either enhancer, enhancer active 1, or enhancer factor 2 were the ones that were providing the best enrichment score for a particular tissue. And if you think that, well, this isn't acting through a variant that's located over an enhancer, and instead this is a case of a change in transcription or something else, CTCF binding, then you can add those. And sometimes they change the result and sometimes they don't. But this is how we've proposed to come up with an interactive tool that will allow you to examine both annotations and the tissue predictions. Now, since this table is built using the same underlying libraries as the other tables that we've added recently, you can transpose it, you can zoom way out and zoom way in. So hopefully the idea here is that as we add additional tools, such as the variant table that I'm going to be working on later this afternoon, that you will have a familiar set of tools to work with. Now, let me look at one more phenotype before I hand back the controls. We have LDSR results and Gregor results for most of our phenotypes, not all. We also have another set of predictions for a few of the traits, and BMI in particular. We have depict predictions. You may remember seeing depict results from the gene table, but we also have depict predictions here. And I know this is using their complicated approach of using co-regulation to put together a mapping between genes and tissues, and so here you have another set of values to look at, and I can sort by them, and these seem a little bit less compelling as far as tissues, but fine. Our goal here is, again, as Maria indicated, to be able to generate lots of computational results, to put them in front of people and let people work with them. For those of you who are experts in particular areas or particular results, you may have a reason to look at a particular computational method, and you maybe ascribe a particular significance to it in a particular context. So this is a new development. There are still some challenges to work out. We need to harmonize across the tissues better, and resolve exactly how we allow the comparison between tissues and cell types. But this is, as always, our goal is to try and get these capabilities out there early so that people can look at them and criticize them and help us to make them better. Okay, so I've spoken my piece about this table. I'll stop sharing unless anyone has any immediate questions about this tissue table. I will stop sharing and then we can do the general Q&A.
So we'll turn over to questions. I saw a question from Melissa, which we'll address right away. What other events are coming up? Well, we'll have a webinar in August. In September for this particular series. We also will have a production release coming in mid-August, which will have new data, new tools, and a new look to the portal, which will be very nice. DK's been working on, the team is really lovely. We'll have new video tutorials, chapter 3 in our YouTube channel. And we'll also have upcoming videos for DGA and our data intake procedures, which I think will be a really good feature. We also are exhibiting EASD in September this year, so it's our first international jaunt with our EBI team. We're going to be featuring EBI as our federated node and the knowledge portal there. And then we have a workshop coming up actually, which will be really fun at ASHG, where we'll be talking about variant to function. We'll have external speakers for that. We'll be talking about what the portal will do in that space. But we'll also be talking about different approaches in the field that'll be valuable in that area. None of this is done without an amazing team of people, which we just added to. Kui, who's sitting on the table in the room here in Cambridge. Wonderful people here locally in Boston that we get to work with to build the knowledge portal and the UI, but also moreover people internationally from all the 4 different sites and many other sites who are just remarkably wonderful to work with, and we're very gifted to have them. I mean, it's a gift to have them in our world. So with that, questions. I saw a wonderful question from Melissa. Let me get it up verbatim. It's in the chat line, but I'll say it for those of us in the room. What she wanted to know was, in the future, could the gene focus table interface be modified to include any user-generated lists of genes, not region-based? This has been a common theme, and this is really a great idea, and I'm getting nodding in the room. And I just chatted with Jason. We think, yes, easily this is something we could do. We will probably do it for October and integrate that into the features that will be allowable in the portal. So that's something we'll definitely do. Thank you for that question, Melissa. What other questions or comments do people have? You know, we showed you a lot of new tools, and I think one of the things we've noticed is that, you know, this requires us to sort of restructure the portal in terms of how we present it, because we have a lot of things in different places that kind of feed into one story. So right now we're building the components of the story, and we're starting to then, over the next couple cycles, sort of bring them together. So we'd love feedback on, you know, usability and workflow, as well as, you know, any scientific questions or portal-based questions. So I'm opening the floor to anybody.
On the tissue enrichment table,
I'm gonna try something.
would it be possible to have another link where you could see the variants that were contributing the most strongly to the scores? So for example, that were contributing to the lower p-values?
Yeah, I think that's a great idea, and it should be displayed. I think that would be a great addition.
Thanks, that'd be super cool.
All right, like I said, this has been recorded, so you can play it back at your leisure. If you're excited about it, we'll have it posted on the site. We'll have our next one sometime in September. We'll send out a date for that. And then look for an August release. And we'll see you at ASHG if many of you are there. As always, like, come by if you're local, email us if you're not, and we will love to hear from you. Thank you, everyone. Take care.
Thanks, everyone.
All righty.